Hello, everybody, and welcome to the Release 65 webinar. I'm Gloria Isabel Giraldo Calderon, and I'm joined today by Evelina Vashenko and Susan Warnfelds. So today we are going to talk about the release news for Release 65. And for this release, we have 3 new features. The first one is the webinars page. So if you come here to the homepage of any of the sites, click on Help, learn how to use ViewPath, and later click here on Webinars. This is our new landing page. At the top, we have organized the upcoming events that we have. Releases like the one today are linked here with the recording that we have in YouTube. And here on-demand training is those specific webinars that we have, for example, this one for the TIC and MITES community, or we have this one about the gene record pages. So we welcome your feedback, and we hope this page helps you to better navigate what we provide. Another feature comes from the OrthoMCL database. So if you have a strategy in which you have different ortholog groups, for example, here one group I have with for kinases and this from ligase, if you scroll to the side, you are able to see the different clades in which your groups of interest appear. But now what we provide is for you the option to download this. So notice here having amoeba, fungi, metazoa, but if you click on download, and you are able to select phyletic distribution clade abundance. So before you were only able to see it, but now you are able to download it as well. And the third and final feature is MapView. This is a tool that is available in Vectorbase. So if you come to the Tools menu, notice we have 2 links for the map. So as some of you may already know, in addition to all the different genomes and transcriptomes and proteomes that we host in Vectorbase, we also have field-based data. So we have Vectorbase more than 1,200 data sets. And some of these are this field data. And the way you can identify them is because you see this MapView accession code here. And for example, these ones here at the top are collections from different mosquito control districts. And this one here says this is a collection from year to 2017, 2016. This one starts in 1998 and goes to 2019. So if I go to the tools, this is the new map, the beta version. And something that I really want to emphasize is that this is just a beta release. We are still working on it, but we want to get your feedback, your early feedback of what can we do better. Just with any other similar map, you can zoom in and out in a different location that you may want. For example, here I'm zooming in a specific country or region, and notice how these markers start to disaggregate when I zoom. Let's give it a minute and see how, and I can keep zooming in on any specific region. Besides zooming on a specific data set or location like that, you can also use the filter option. And here what it's telling you is that we have 738 studies, and we have these collection sites, the collection samples, and this is how the data is organized. So we have insecticide resistance, phenotypes and genotypes. We have population abundance. We have pathogen infection status. If an arthropod mosquito is infected with a parasite or a virus or a blood meal source. You can start to select the different filters from here and say, oh, I want, instead of species, which is what I have here, I want to display the sex of the samples that I'm interested. I only want to see females, for example, and notice how the filter now appears here at the top. You can download the data sets. You can download everything or just download what you have been selecting on the screen. You can share, for example, you select a marker, you want to know more about that one, and you create a filter on your screen and you share that. And you can type here notes of what have you done. Oh, I went to US, I select specific city, I want this species with this sex. And on My Analysis, you're able to see all the queries that you construct. And if you're visualizing a specific data set, you are able to see later details about that data set and who collected the data or any type of details that you may want about this analysis. So what does people do with the map? Well, when they publish with vector-based map, they gather all that metadata that we collect, the latitude and longitude coordinates of collection site, the collection date, trap type, attractant, abundance, or any other fields. We have between 20 and 25 different fields that people can use. So you can either use the map interface or download the data to perform your own analysis. For example, this is a paper published in Science in the year 2020. From different sources, including vector-based, they compiled these long-term surveys to talk about, insect decline abundances. In this other paper from Trends in Parasitology 2021, they were using our blood meal data to work on the host insect interactions, host disease interactions. And this other paper from Science Reports from this year, so they download the data exclusively from vector-based, and they said, okay, about 7 species of interest we want to see in a time of 20 years, how is there different abundances? And not only people publish reviews with the vector-based map, but they also say, okay, this species has recorded for the first time in a different location, or how the abundance of our species correlates with both biotic and abiotic factors. So those are the kind of things you can do with the vector-based map. We hope you enjoy the map. Remember, if you're going to publish right now and do your analysis, we recommend you work with the old map, because remember, we are in beta version right now and still working on it, and you may get some bugs as you work on the map. Okay, now what we have in vector-based and about genomes, we update our databases every 2 months. And for vector-based, we're right now strongly working, especially in the Anopheles and tick species. And some species, we changed the genome version for a new one that has previously many supercontigs and now just has genome organized in chromosomes, or we changed the name of the strain. So to provide a little easier for you to know what we have changed, we created this table. And here we provide a little bit more of details about these changes. For example, for this Aquasalis, Colusi, Funestus, Amoucheti, Anopheles, we changed the name. For these names that were long and not really human readable to some things that are probably easier for users to remember. So the strain names have been changed. And in the case of Drosophila, we just updated the genome version and now provide a mitochondrial genome. Something very important for this release that it has been first tested in ticks, but we'll also share in other databases, is that for some gene products, you may have noticed that they had just hypothetical protein or unspecified product, things that were not really useful. So now we have PFAM domains have been substituting this uninformative information and just giving you, better description for the gene in question. And this, as I said, will impact many other genomes in all the ViewPath databases, not just vector-based. And for vector-based in this release, we have 2 RNA-seq data sets, one for Aedes aegypti and one for this tick, Hemaphysalis longicornis. So now we give pass to Eve, if there is now any questions about what I have shown about vector-based.
Right, I'm setting up my screen, so you should be able to see the FungiDB news release.
Yes,
yeah,
okay.
That looks great.
All right, thank you, that's great. So I will talk briefly about the new data sets we have in both FungiDB and also hostdb.org. Notice that I've navigated to the sort of landing page for the release 65. but you can, where you can also view other releases if you scroll down, if you want to catch up on the history of the releases. So just in general, in FungiDB, we have integrated several new genomes. These genomes span different genera and species that can be classified as plant pathogens, human or animal pathogens. For example, we have integrated Fonseca multimorphosa, which is a fungal organism that causes infections of the central nervous system. We also have an agent that causes histoplasmosis, and the W24 strain is a clinical strain that recently have been sequenced and annotated. We have also integrated a assembly and with annotation for Martorella mastotomatis that causes mastotoma, which is a chronic and progressive disease that can spread to different organs and tissues in humans. And we have also integrated additional genomes that are important model systems. In terms of the functional data sets in FungiDB, we have quite a few data sets that span transcriptomics and also SNP analysis data. And first, I'll just briefly overview what we have for transcriptomics. In terms of RNA-seq data that we have integrated, we have several data sets for Candida species, such as Candida albicans, Candida auris, and also Rhizopas delamar, which has made news as a COVID-19 infection that was popping up in various clinical conditions. So these three data sets, one for Candida auris, one for Rhizopas delamar, and also one for Candida albicans, the third one, Transcriptome Dynamics in Canada, are also associated with a host data. That means that the researchers have used RNA-seq to characterize basic transcriptomes, whether it's characterization of the initial stages of the infection or general fungal host interactions, and we have integrated this data. The fungal aspect of this data is available in FungiDB, while the host component is available on hostdb.org. So if you are interested in host pathogen interactions, you can navigate to both of these websites and, for example, click on the link associated with one of the data sets. The link that is highlighted in blue within the news record page will redirect you to a dataset record page. The dataset record page provides additional information in terms of the publication that is associated with it and also how you can mine the underlying data. Notice that for this particular dataset, we have several options available, such as fold change, percentile, and you can also look at the sense and anti-sense transcripts. This particular option becomes only available for the datasets that use strain-specific kits for DNA extraction and subsequent sequencing. I won't go through the details of how to run the analysis, but the idea is that you can click on any of the provided links, navigate to the samples that are associated with this data set. For example, Candida albicans was studied when blood from several human donors A through C, were used in the study, and you can look to see any similar or sort of different trajectories in terms of gene expression when it comes to the host pathogen interaction. And so, as I have mentioned before, you can mine the host data within the HostDB, while if you want to mine the fungal data, you would navigate to FungiDB and essentially carry out the analysis there. Now, for the SNP analysis data sets, we have integrated whole genome sequences from several projects. These sequences come from different studies that are associated with Aspergillus fumigatus, which causes aspergillosis and is quite common in infection, especially in immunocompromised people. We also have integrated SNP datasets for Candida auris. I have already mentioned Candida auris, but Candida auris is an emergent and probably already established pathogen that causes severe disseminated infections in clinical studies. And the interesting thing about this pathogen that it has acquired multiple antifungal drug resistance phenotypes. So, for example, for Candida auris, we have 3 data sets that look into basically SNP calling in isolates that were collected either by the Nevada State Public Health Laboratory or during the outbreak in Italy, or the CDC project that collected multiple clinical isolates of Candida auris in the US. We have also integrated isolates for Cryptococcus neoformans that causes also fungal infections in humans and in animals. And you can explore all of these data sets using SNP-centric or sequence-centric searches within FunGTB. For example, if I click on one of the links within Candida auris list, again, I'll be navigated to the dataset record page. So you could scroll down the dataset record page to identify different types of searches that are available for this type of data. In this particular case, you may be interested in the SNP characteristics. For example, we know that isolates, they're under pressure to evolve a antifungal drug resistance are often associated with increased rate of non-synonymous mutations. So for example, if you pick Candida auris dataset from the drop-down menu to adjust the parameters and the selection criteria for this particular organism, you can click on the dataset to explore all of the datasets associated with this organism in FunGTB. And for example, if you want to interrogate a specific dataset, you can just select this one, or you can select multiple datasets if you choose to do so. You can also use the various metadata associated with the sample collection or just the sample sources to essentially use the metadata criteria based on the health status of the patient rather than individual dataset selection to run your analysis. And I won't go through all of the details of the search, but if you scroll down, notice that the SNP characteristic search has a SNP class parameter that you can choose to specify non-synonymous. And so the idea is that if I deploy this search and I'm using all the default settings at this time except the SNP class, if I click on the get answer, my search will run the analysis to return non-synonymous SNPs that are identified within a specific data set that has been generated by CDC. And so once the page refreshes, you essentially will have a first step in the strategy that you can explore within FunGTB by adding additional steps and bringing any other types of data. So let's see. And so here's the page refreshed, and there are over 5,000 genes that correspond to my search criteria. And so notice that you will have a results table underneath your search strategy with additional information in terms of the SNP statistics, etc., that may be useful for you if you're looking at a specific group of genes. Right, and so this is for me in summary, and at this time I will turn this over to Suzanne.
Okay, thanks. Let me see if I'm presenting. Do you guys, okay. Do you guys see a TrickDB?
Yes.
Okay, awesome. Okay, so I'll describe the data that's in the parasite sites, which there's new data in TrickDB, TriTrickDB, ToxoDB, PlasmoDB, and Crypto. So I'd like to tell you about that now. And so we're looking at the homepage for TrickDB, and if I go to the news, I can see that we had one genome update in release 65 that T. vaginalis G 2022 was updated to the new version at GenBank, right? So that has been updated. And if I go to searches for organisms and look for, I can get all genes, right? And I find that there's 88,000 genes. We also have another useful search if you want to go to gene models and gene model characteristics. I can choose only this new version of T. vaginalis G3 2022 and look for, say, all transposable element
Hi,
genes.
Suzanne.
We are losing your voice, Suzanne. It seems like your volume is going up and down. Is that happening to you too?
Yeah, the same. You seem to, in sort of sprouts, it's almost like you have two microphones and one is picking up versus the other.
Oh, that's weird. I can switch microphones. It'll take me a few seconds. I'm sorry. All right, so I'm using the gene model characteristics search to look for only transposable element genes in this Trick vaginalis. It's known to have many of those. So although we have 88,000 genes, 35,000 of them are known as transposable elements.
No, seems like we still have the same issue, Suzanne. Sorry.
Yeah, I'm sorry, I don't know. Okay, is that any better? I don't.
Yes, you sound louder. Good.
So far, so good. Yeah.
All right. So I guess I'll just go on to TriTripDB. I'm sorry about the sound. I really don't know what's happening. But in TriTripDB, we have, several new data sets. We have some hyperlopit mass spec data, and then our annotators have updated the 927 genome with 171 new annotations for different genes. And if you are interested in those, they've also created a Google Doc that you can access that chronicles those changes. And your comments are actually reviewed, right? We're happy that you all are adding comments to genes, and we've integrated some of your comments into the these data sets. But I'd like to talk a little bit about the low-pit mass spec data, that which is a quantitative mass spec technique method used to separate organelles and other subcellular compartments. So the data is really, really interesting. So I'll just go to one of those data set pages. And this is a published data set in Nature Communications in 2023, so it's relatively new data. And we have two data sets, like they looked at the bloodstream form and the procyclic form parasites in both of these to find the suborganellar compartments that are where genes are located, right? So if I can say, I can go to that search for genes and look for low-pit. And of course, you can do this search all by itself, just to look for genes if you're interested in genes that are, possibly in the nucleus. They say there's 310. We use our filter parameter here, as we use in many other searches, especially the SNP searches like Eve demonstrated. But you can also use this in combination with other searches. So if I want to start a search strategy, I could look for, say, genes that are upregulated by RNA-seq data. So I'll use this search adipose tissue versus bloodstream form and look for genes that are upregulated in the blood versus fat. Yep, and I find about 1,000 genes that are upregulated in blood versus fat in this published data set. And then for those, I can look to see where those are expressed in subcellular compartments. So if I intersect that data with the low pit, that result with a search for genes that are in the nucleus, as evidenced by this low pit data set, I find that 30 of these blood-related genes are actually in the nucleus of the cells. So it's a pretty good, very interesting search to use in combination in our search strategy. I'll move on to ToxoDB. And in release 65, we had some annotation changes in Sarcocystis neurona. This is one of the genes that had one of the genomes that had greater than 90% uninformative product descriptions, such as unspecified product. And we went in and looked to see if the genes were annotated with PFAM domains, and when they were, we substituted those PFAM domain names for their uninformative product descriptions. There's a little search here that we did that we can show that shows that about 3,100 genes were given new product description, and those are their PFAM domain names. So if I go back to the news, we also updated this bulk RNA sequence data set. It was originally loaded in release 54, but we were, because of an error that we found elsewhere, we were a little bit worried that this might have an error in it. So we updated that and we found that there was no error, but we're letting you know that that we reloaded it. And then we also loaded this single cell parasite transcriptional atlas of T. gondii RH. This is actually a sister data set to two that were loaded in the last release. So if I come down to associated data sets, there are two other data sets within ToxoDB that are in the same publication as this Toxogondia RH data set. And of course, well, I'm going to talk a little bit about more in depth about the single cell data when I get to CryptoDB. So if I go back to the news, we can also see that our annotators have updated 176 genes in T. gondii ME49, and there's also a Google Doc here that you can look at that chronicles those changes. Okay, in PlasmoDB, if I go to the news, we see that we have added 8 data sets to this whole genome sequence patient isolates from Peru. So we were keeping an eye on the sequence read archive that contains these whole genome sequence data. And we noticed that the authors of this paper added data to it, and so we have uploaded and analyzed that data also. Of course, you can access this data through all of our SNP searches, but I wanted to demonstrate something interesting on the gene pages that maybe you don't know about. So I'm going to go to a gene page for, I don't know, let's say glycotransferase. And this data is associated with P-Vivax. So I'm going to limit my site-wide search for glycotransferase by the PVIVAX genes, and I'm just going to go to the first one here. So if I go to the SNP section, it will load an image of all of the single nucleotide polymorphisms for this gene. But if I scroll down, we have another tool here called isolaalignments in this gene region. So, and we have our filter organism here. I can choose my data sets, whole genome sequence of patient isolates from Peru. And I can ask it to show me an alignment for these 16 isolates in the region of this gene here. I'll navigate back. So I'm going to go ahead and use this tool to look at an alignment of these 16 isolates in the region of this gene. I can say view results, and although sometimes this can take a while, so I've actually already done that here. And the results show us what different isolates we've used, and then show us an alignment of these genes, which you know, you can look at here, or you can copy and paste into other resources to look at that. We also offer a link to view the phylogenetic tree alignment, and that goes to ITAL, which allows you to investigate this a little bit further. Okay. Let's see. Yeah, that was all that was in PlasmoDB. And if we go to CryptoDB, we have a little bit more data there. I'm going to go to the news. We have a single cell RNA sequence that is provided by the Streepen lab, and it has not yet been published. They are working on that, but they thought that we would be able to show this to our users. And I'm going to demo that in a minute. And then we also have two genomes, two organisms, Perospora gigantea A and B, which went through our PFAM domain to product description. And so there's more information there if you're interested in comparing these genes in these organisms to the ones that you're studying. So I'm going to take you to the search for single cell RNA-seq and demo that. This is just a simple search that will return all genes in a certain experiment, right? So all of this data was aligned to Cryptosporidium parvum Iowa 2. And I'm going to look at this asexual stage atlas and ask get answer. And so I find that there's 4,000 genes in that experiment that have data for this organism. Now you can access, or for this experiment, you can access this data on the gene page, right? So let's just go to a gene page. Let's choose one that has So here I am at the gene page for this signal peptide region containing protein. And I'm going to use the content section to go down to transcriptomics. And we have a new table here called single cell RNA-seq. It's similar to our other tables where each row is a different data set and you can expand those to see details. Right, so here we have an interactive portion of the cell by gene app, and you can, look for different expression values here and see where they show up. Those what cells they are expressed in the UMAP. You can also use this link here to open up cell by gene for this particular gene. So this is a UMAP of this showing the expression of this particular gene, GD1-130. So over on the left here, we have metadata associated with this experiment, so you can explore different time points or stage of the parasite. You can color each of these by that, and you can also, like, say, do a differential expression. So if I want to say that I have male cells that were identified as male gametocytes, and I add those to one data set here, and then if I say I have 2,500 that were identified as female, and I add that here, then I can use this little button to show differential expression. And I have 2 gene sets that come from that. One gene set is high in my males, population 1, and one gene set that is high in my population 2, which was my females. So there are tutorials online if you want to delve deeper into cell by gene and how to look at that. But I just thought I'd give a quick demo of that. And I think that's all we have for you today. I don't know if there are any questions.
I don't think there are any questions so far.
Okay. That sounds great. Well, if you do have questions, you can use the Contact Us link on any of these sites, and you could email us your questions. We try to get to those within 48 hours. And if we don't, just ping us again, just send another email, and we're happy to talk about any question or suggestion that you have. Nothing is too small to bother us with. All right. Take care. I hope you guys have a, yeah, thank you. Thank you, Juliana. All right. Y'all take care.
Thanks, everyone.
Thank you.
Bye.
Bye.